Good afternoon, everybody. First, I would like to thank the HBL committee for offering me the opportunity to present my work. Today, I will talk about my research on the whole brain mapping of somatosensory responses in awake marmoset investigated with ultra-high field functional magnetic resonance imaging. The somatosensory system is organized in a similar way in a wide range of mammalia species, such as rodent, cat, or primates. Two systematic representations of the contralateral body surface have been found. One primary representation in the primary somatosensory area called S1 in red, and a second representation in the second somatosensory area called S2 in pink. Dermis of the skin. This receptor receives stimuli from the outside world, such as heat, pain, pressure, or cold. Interestingly, adjoining groups of neurons across the surface of the cortex are activated by different and typically nearby body regions, so that all parts of the body are represented in a pattern relative to the cortical surface. Why use marmoset? The common marmoset is becoming an important model in neuroscience and for translational work. Indeed, marmoset and humans share similarities such as social relationship, behavioral abilities, and social cognition. Particularly, this primate has a small size, allowing an easiest handling compared to macaques, and the possibility to image the brain at the whole brain level with a high resolution. Thanks to this high resolution, we're able to see subcortical activation, which is difficult to obtain with macaque brain. Lastly, his smooth cortex, so it means no gyri or sulci, allows for laminar electrophysiological recording. The somatosensory system has been studied for several decades in numerous mammals, including the common marmoset. Like humans and other non-human primates, both S1 and S2 have a somatotopic representation in marmoset. This means that each body part activates specific region in this area. So for example, the face activates this part, the hand this part, and the foot this part. However, all of these studies were performed under anesthetized condition or using postmortem analysis. Recently, Dr. Alfonso Silva and his team successfully acquired functional MRI in awake marmoset. They explore the difference of brain activation in response to somatosensory stimulation applied to the wrist between awake condition and anesthetized condition. They observe significantly attenuated response in S1, S2, in subcortical areas such as the thalamus and the caudate in anesthetized condition compared to awake condition. So these results suggest that the somatotopic maps obtained in anesthetized monkey may underrepresent the reality of the tactile processing occurring at an awake state. As such, it's important to measure the somatotopic representation in awake marmoset. The aim of this project was to establish the feasibility for getting high quality and reproducible sensory motor mapping with fMRI acquiring awake condition, and to explore the functional body representation by applying tactile stimulation to the face, arm, and leg of awake marmoset, and compared to studies performed under anesthesia. Lastly, we also want to explore how somatosensory areas highly responsive to tactile stimulation are functionally connected to some cortical and cortical region. For our task, we use the pneumatic Galileo somatosensory system to deliver air puff to the face, arm, and leg of the animal. So we first shave the arm and leg to tape a stimulus node around them. The lower part of the face, encompassing the nose, mouth, and chin, were stimulated by a plastic manipular arm put in front of the animal face. AirPuff were delivered in a block design. So face, arm, and leg were stimulated at a frequency of 5 Hz in different blocks, separated with non-stimulation condition. We acquired data in 4 marmosets: face, arm, and leg. Stimulation elicit brain activation in the contralateral hemisphere of the stimulation. So here it corresponds to the right fiducial hemisphere of the animal because we stimulate the left body part. Face activation are represented in red, arm in blue, and leg in green. Particularly, we found activation in S1 and S2. In S1, the face activation are found in the lateral part, the arm activation in the middle, and leg more in the middle part. In S2, face activation are found rostrally compared to the arm and leg activation. As the somatotopic organization looks similar across monkey, we then perform analysis at the group level. With a total of 64 runs, we obtain this figure which highlights a mediolateral progression from leg to face for S1 and the rostrocaudal progression from face to leg for S2. Subcortical activation in the thalamus and putamen shows some somatotopy mapping too. This somatotopic organization follows a mediolateral progression from face to leg in both subcortical areas. These results are consistent with previous tracing study performed in the thalamus of marmoset. However, this is the first study showing a somatotopic organization within the putamen in marmoset. A similar body representation has been shown in old world macaque monkeys and humans. So this result suggests that this is a conserved feature across primates. To go deeper in our analysis, we define 6 volumes of interest in S1 and S2 based on the group level analysis. These voids correspond to the face, arm, and leg activation local maxima in these areas. So we then use resting state functional data to compute the functional connectivity maps from the 6 volume of interest with the medial frontal cortex. We observe strong connectivity between voids defined in S1 and the posterior region, and with mid-cingulate areas for face and arm voids. Voids defined in S2 show strong connectivity for both posterior and mid-cingulate areas for face, arm, and leg. For the mid-cingulate areas, the hotspot of connectivity was more anterior for face compared to arm and leg, suggesting a body representation mapping within the mid-cingulate areas. Interestingly, a somatomotor map for face, upper, and lower limb has been identified in rostral cingulate motor areas and more posteriorly, both in human and monkey. So our results suggest that a similar organization is present in marmoset. So with this project, we show a strong contralateral functional activation related to each body part in S1 and S2, and that these activation areas are generally consistent with previous findings obtained with invasive method. We also examine the subcortical activation pattern associated with tactile stimulation and highlight that the thalamus and, for the first time, the putamen exhibit somatotopic pattern in marmoset. Lastly, using resting state functional data, we also reveal that S1 and S2 exhibit a body representation in their functional connectivity pattern with the posterior and mid-cingulate cortex. This activation and disconnection were consistent with previous findings in human and macaque monkeys, indicating that the organization of the somatosensory system is conserved across primates. I would like to thank my supervisor, Stefan Everling, and all the persons involved in this project, as well as my team members, and of course, BrainSky for the funding. Thanks for your attention.